Today my talk is titled Global Patterns in Stream Energy and Nutrient Cycling, and I am your host, Adam Wymore, and reporting today from a very snowy New Hampshire. But any working group or any collaboration or synthesis effort, like what's happening, is in no way a solo endeavor. And so it's very important that right in the beginning to acknowledge all the other working group participants who have committed to this collaboration and are working to push things forward. But I also want to take a very special moment here to acknowledge the NC's data team, and especially Julian Brun and Margot Sleckman, who have been instrumental in helping in this process, synthesizing this amount of data from these large, diverse data sets is no small undertaking, and their help has been instrumental in keeping this project moving forward. And anyone that may be in Santa Barbara right now doing their own working group, I encourage them to work early and often with the NC's data team. Their expertise is invaluable. So a big thank you to all the members of the NC's data team. So first thing I want to do is provide a bit of an outline for my talk this afternoon as we look at a couple pictures of different LTR sites. First thing I want to do is talk a little bit about the value of long-term observational stream chemistry data and what we can do with those types of data sets. I'd like to introduce the working group and our overarching objectives and questions, and then share some of our initial results and insights, what we're learning from this large synthesis effort. The focus today is primarily going to be on carbon and nitrogen interactions. We'll look at the scaling properties of dissolved organic matter, and because of the structure of these data, we'll be able to work across scales. And then at the end, talk a little bit about future possibilities, where the working group is going to go, and potential other collaborations that exist. And I'd like to begin the kind of formal part of the talk with one of my favorite quotes from H.B.N. Hines in a lecture that he gave titled The Stream and Its Valley. And he reminds us to look at streams not as purely aquatic phenomenon, but rather to view them as parts of the valley that they drain. And so what you're seeing in these pictures here is the small picture here. This is Oak Creek Canyon in northern Arizona, draining the southern edge of the Colorado Plateau. And for the last 8 million years, the little stream of Oak Creek has slowly carved out this beautiful little canyon. Now sits at the bottom of this canyon, processing everything that comes out of this watershed. And so what that means is that long-term observational data sets are highly valuable for both stream ecology and watershed and ecosystem science. And this is because streams integrate the watershed, and because of hydrological connectivity and gravity, things move downhill and into the stream, and therefore streams are excellent indicators of broader ecosystem and landscape scale processes. And I'm going to give you two brief examples. One is first from Hubbard Brook, and that it's through nitrate concentrations in the stream, here on the y-axis, that we can detect the successional stage of the forest through changes in rates of biomass accrual. We can also find the signal of photosynthesis turning on and off within the stream chemistry. And from the Keo LTR, which is also a critical zone observatory, we can detect how the structure of what we call the critical zone, so from the weathering front all the way to the top of the vegetative canopy, how the structure of the critical zone, represented down here in this principal component, how it influences the concentration discharge relationships of different solutes. And you can see in this case of sulfate and phosphate. That is, as your bedrock, your lithology, and the composition of your forests change, well, so do the concentration discharge relationships. So again, streams integrate the watershed. But at the same time, and very importantly, streams are also biological reactors driven by microbial engines. And so what you're seeing here in this figure on the left is a classic figure from Pat Mulholland's 2008 paper, which is the microbial process of denitrification varying across land use within the stream. So a key ecosystem process within the stream. So the take-home message from this little intro is that stream chemistry is an integrated signal. And what it's integrating is meteorological, biological, soil-based, hydrological, and geological processes, all within the stream channel. And this is both a kind of a blessing, but at the same time a limitation. We can learn a lot from stream chemistry, but we have to be careful too at the same time and recognize this limitation as from observational data sets, it can be hard to test direct mechanisms, but we can sure try. And so this is where the LTR program comes in. And what the network of observatories offers us is multiple long-term data sets to synthesize stream chemistry data across large environmental gradients. And if we just briefly look at the distribution of LTR sites across the continent, even though we're not necessarily using all of these right now, but we can span from the tropics down in Luquillo in Puerto Rico all the way up to the taiga in Alaska. This is part of the power of the program that was set in place decades ago, in which we're now able to tap for these large synthesis efforts. So on that note, I would like to introduce you to the working group and this team of collaborators. Diversity exists in many ways. One thing that makes this working group particularly effective is the different ways that we look at watersheds, whether we're freshwater ecologists, or modelers, or geochemists, or hydrologists, or Bayesian statistician. It's this diversity of worldviews that allows us to do different things and to take data and look at it in novel ways. But we're unified around biogeochemical cycles. And in particular, dissolved organic matter and dissolved inorganic nitrogen. I think there's a future within our working group for more incorporation of these other solutes, but this is where we're really focused on right now, is these interactions between carbon and nitrogen across these environmental gradients. And so this sets up our overarching objective of the working group, which is to develop a more complete framework for understanding how the cycling of dissolved organic matter and dissolved inorganic nitrogen is linked across environmental gradients. And to make a couple points about DOM and DIN, we know that DOM, dissolved organic matter, is both a major energy and nutrient source to stream ecosystems. This duality is really important when we talk about DOM. We also know that major transformation in the nitrogen cycle require carbon as an energy source. And so therefore, because of points 1 and 2 here, the biogeochemical cycling of dissolved organic matter and dissolved inorganic nitrogen is inextricably linked. We can't, and to understand one, we have to understand the other. And developing this framework is really important at the global scale. And I provide a couple examples here. This figure is adapted from Spencer et al.'s 2013 paper in GRL. And the point I want to make here is that the amount of carbon stored in dissolved organic matter is roughly equivalent to the amount of carbon stored in the atmosphere as CO2. And so we're talking about a globally relevant pool of organic matter. And you can also see that on the annual basis, the continents are exporting a lot of dissolved organic matter. And these are some of the major watersheds here on the North American continent. And I want to draw your attention to the values that we see here at the Yukon River Basin. And understanding dissolved organic matter in a changing world is quite relevant because of the changes that we expect to happen in these permafrost landscapes, that these values could change over time. At the same time, nutrients, including nitrogen, have very important consequences for water quality and eutrophication. And I want to use an example here out of our backyard in New Hampshire. This is the Great Bay Estuary, where all the streams and rivers in our little corner of southeast New Hampshire drain into. And the Great Bay is nitrogen impaired. And it is currently in violation of the Clean Water Act as a result of that. And you can see in this figure here on the bottom that it not only is it dissolved inorganic nitrogen that contributes to this nitrogen load, and this is primarily in the form of nitrate, but that organic nitrogen is contributing a significant portion, the green bars, to the nitrogen load experienced within the Great Bay Estuary. So these are really fundamental questions that are relevant to global biogeochemical cycles and water quality. So that sets up our overarching question. And that is, under what environmental conditions are the concentrations of DOC and DON correlated with the concentrations of nitrate and ammonia? We can look briefly at to the literature for some generalizable patterns. And a lot of people have probably seen figures like this or similar in which we see this dramatic negative curlinear relationship between DOC and nitrate. And this is the top figure from actually Helen et al. Is a synthesis of USGS data to establish this pattern. At the same time, we see experimental evidence that corroborates this pattern. These are data from Hubbard Brook, and you can see with the addition of acetate, a labile form of carbon, that nitrate concentrations are drawn down quite low. But at the same time, while we see these patterns, we also see exceptions and inconsistency across studies. This figure and these data are from Bianca Rodriguez Cardona's paper. This came out of her master's degree. She's currently a PhD candidate at the University of New Hampshire and a member of the working group. And what she's done in this figure is on the y-axis is she has microbial uptake efficiency plotted against ambient DOC concentrations on the x-axis across studies here. And you can see that there's no significant relationship between DOC and this important ecosystem process. We see important interactions between nitrate and DON, for example. This figure is from Brian Peller et al. We can see that as watershed DIN loads increase on the x-axis here, that DON concentrations increase, suggesting an interesting interaction between two different sources of nitrogen. But at the same time, there are also inconsistencies. We see temporal variation in this interaction between nitrate and DON. In the summer, we see a significant positive relationship, but in the spring, a significant negative relationship. And from an urban system, in particular the Baltimore LTER system, we see the same type of variability. In spring, there's no relationship, but in summer and fall, we have significant negative relationships. And there's even less evidence for a generalizable DOC ammonium pattern. So this kind of leads us right back to where we began here in this question, with a big question mark. And we hope that with a new synthesis of data and new ways of thinking about these types of data, that we may be able to uncover a new understanding of how these solutes and how carbon and nitrogen interact in stream ecosystems. Well, on the topic of carbon and nitrogen interactions, I want to acknowledge that there's one more interaction here, and that's the interaction between DOC and DON, the two fractions of DOM. Something we're learning more about in recent years, and another way that carbon and nitrogen interact within ecosystems. So what our working group has set out to do, and has done to a certain degree, is that we are synthesizing multiple long-term observational stream chemistry data sets to examine this interaction between carbon and nitrogen across environmental gradients. And this map shows you the current gradients that we have covered. The red stars represent the LTR sites that we've currently incorporated. So these 7 sites, and again, just these 7 sites alone get us from the tropics to the northern deciduous forests, tall grass prairies, the coniferous forests, all the way up again into the taiga. But what's been particularly fun and exciting about this working group is the generosity of others to contribute more data. And that's what these blue stars represent. And so we have a list of contributed data sets. And I should acknowledge that the stars are just sites. Usually under each star are multiple streams and watersheds within each area. And so because of the contributed data sets that we have, we now have replication across the Arctic through the Arctic Observatory Program. I want to make a special point to this data set that was donated across a north-south gradient in Finland, which is over 40 streams and rivers, some of them with over 40 years of record. Absolutely remarkable data set. We have data from here in New Hampshire across a development gradient, agricultural data. So this has become a really exciting prospect to be able to have this type of data in hand. And what that means is that our data set goes from the tropics to Arctic, We have headwaters to now some of the largest global rivers. And we cross from pristine to agricultural to urbanized gradients. And at the same time, this represents collaborations across observatory programs, which is particularly exciting. Of course, this is a large effort through LTER, but we have efforts now, observatory programs, monitoring the Great Arctic Rivers, critical zone observatories, NOAA, among others. So cross observatory networks is a very powerful way to increase our environmental gradients. So what can we do with this kind of data set? This is when things I think get really exciting. Well, one is we can begin to describe and quantify the variability of stream water chemistry at the Earth's surface. And in and of itself is exciting and very meaningful to start to be able to put some bounds on this variability in concentrations of solutes. Second, we can detect patterns at multiple scales as a result. We can work at individual streams, we can work in regions, to biome, to global. And third, we can generate testable and competing hypotheses regarding mechanisms which control biogeochemical cycles and elemental fluxes from watersheds. And again, this gets to a bit of the limitations of observational data, but also kind of points to the wonderful things that happen when we start pairing observational data with experimental data to get to a deeper and more complete understanding of these interactions. So the initial questions that this group is asking are very focused on carbon and nitrogen, like I've indicated. And the first question that we're asking, that we're going to explore here in a minute, is how does the composition of the TDN pool change with DOC concentration? And I want to highlight here this portion of it, the composition of the TDN pool. The figures I'd shown previously in the earlier slides are looking at carbon, DOC, and one particular solute, whether it was nitrate or ammonium, for example. But we're going to borrow something from the toolbox of soil scientists to understand how the composition of the TDN pool changes with DOC concentration. The second kind of overarching question that we're going to ask is how variable is the relationship between DON and DOC? And this is a relationship that's been explored much less, but is getting more attention in the literature as we're starting to understand some of the reactivity of dissolved organic nitrogen across ecosystems. So here we're going to ask specifically, what is the global scaling relationship between DOC and DON? Are these scaling properties invariant across space and time? And the variability in DOC-DON stoichiometry, what is the variability, in DOC-DON stoichiometry, and what is the role of biology potentially in contributing to that variability? Okay, so let's start looking at some data. So again, our first question here is how does the composition of the TDN pool change with increasing DOC concentrations? So what we're looking at here is a ternary plot, and in particular, this is the global scale ternary plot from our data synthesis. And for those unfamiliar with a ternary plot, this has a history in soil science, as it's often a way to look at the contributions of sand, silt, and clay into a soil sample. But what we've done here is our different corners represent the different components of the TDN pool. So nitrate, dissolved organic nitrogen, and ammonium. The axes are relative abundance. And as you go towards a corner, and this one in particular, that TDN pool is comprised of relatively more nitrate. As we go down this left-hand axis, the relative contribution of dissolved organic nitrogen increases. And as we go across this bottom axis, the relative contribution of ammonium increases. Each point within this ternary plot is a site mean. And each datum has a sum of 100, since we're dealing with relative abundance data. The color ramp you're seeing here is DOC concentration. So by doing this method, we can now see how the composition of the TDN pool changes with increasing DOC concentrations. So the first observation we can make is that the stream world is primarily comprised of DON and nitrate. And second, that there's no large contribution of ammonium across sites. Well, this may not be particularly surprising to a lot of people. These systems are mostly oxic, and nitrification is readily happening. But I think what this does is change the way that we may think about how these different solutes are interacting. In particular, what I mean by that is what I want us to notice here is that the reactants needed for mineralization down in this corner are abundant. And the products of nitrification, nitrate, are relatively abundant here. So what I believe this suggests is that ammonium, although fleeting, is a currency of transfer dictating the composition of the TDN pool. And we also see from previous slides that I showed and also other work going on that there are interactions between nitrate and DON. So we're moving up and down this axis in ways that we don't understand yet, really dictated by this currency of transfer that is ammonium. And the last observation we can make of this ternary plot here is the relative contribution of DON to the TDN pool increases with DOC. Now again, a first look at how, at the kind of the global scale, how TDN changes with increasing DOC. What we'd like to do now is kind of scale down a little bit and ask, what is the heterogeneity of this ternary plot as we start moving across space? So what we're going to do now is scale down and look at ternary plots from the individual LTR and long-term sites across the environmental gradient that we have available. So what you're seeing here are 7 ternary plots. The axes are all the same. Corners are still the same with respect to solute, relative abundance on each axis. And here's, again, the color ramp of DOC concentrations. And you'll see that for the most part, many parts of the world and across these biomes, which are labeled here in the upper right-hand corner, the world adheres to this left-hand axis of nitrate and DON. Below each ternary plot, I've also labeled the LTR, the long-term site that we're looking at in particular. You'll notice here in this large finished data set that we actually start moving towards the ammonium corner. We believe that this may be a signal of urbanization. We'll see that in some figures coming up, why I think this is the case. The northern deciduous forest here, which is a combination of the Hubbard Brook data and the Lamprey Observatory data, adheres tightly to that left-hand corner. But what's interesting here is, I'll call your attention to the ternary plot G down here in the bottom left-hand corner. This is from an urban site in San Juan, Puerto Rico, the Rio Piedras. And you'll notice that the composition of the TDN pool here in this urban site has basically shifted to the right-hand axis across the ammonium and nitrate axis. Now, this is a heavily impacted system receiving a lot of urban and waste inputs. So I think what we're seeing here is the ability for anthropogenic inputs into the system to overwhelm the ability, basically, for nitrate or ammonium to dictate that composition along that left-hand axis. We can also see gradients of development changing the composition of the TDN pool. Again, so this is a development gradient here in southeastern New Hampshire. We can see these green points down here are heavily forested and wetland systems sitting in that DON corner. The red points here, which are buried a little bit, these are the highly developed system with a lot of nitrate inputs from septic tanks. But then we have this blue area here that's a moderately developed. So both forest and wetlands are in these systems, as well as the development in the septic tank system. So again, what we're seeing here is the interaction between land use dictating how the TDN pool looks. We see a very similar type of interactions going on in this large data set we have from the United Kingdom across an agricultural land use and different livestock. We can see that depending on the land use and the livestock, that we move up and down this axis and controlling the composition and the ratio of nitrate to DON. So I think what these ternary plots do is they help us start to think about the nitrogen cycle in freshwater ecosystems a little differently. And I want to illustrate that is we have this ternary plot considering all 3 solutes. And I want to go back to a classic figure that many people know from the Peterson et al. Paper in 2001 that really nicely illustrates the nitrogen cycle, but is heavily focused on interactions within the inorganic nitrogen pool, ammonium and nitrate. And by thinking about TDN in that ternary plot framework, it actually forces us to think about stream nitrogen cycling in a more integrated framework. And what I want to share with you is an adaptation of this figure that my friend and colleague Susanna Bernal produced, in which she has adapted the Peterson et al. Figure, but you'll notice that she's incorporated dissolved organic nitrogen into this figure now. So now we have a more integrated framework here. And the other thing I think we need to acknowledge, based on what we're learning from the ternary plots, but other experimental data that's occurring, is there's this interaction between nitrate and DON. It is highly variable. So what this does now is it takes us out of a kind of a linear way of thinking about nitrogen cycling in streams. It actually begins putting feedbacks within this diagram here, showing how we can produce the initial reactant of dissolved organic nitrogen from the end product of nitrate. And then by thinking about those ternary plots against gradients of DOC, we can ask ourselves, well, how do things change in low DOC, low energy systems versus high energy systems? How do the width of these arrows change? And what does that mean for DIN or DON exports? So again, this is this new framework that I think is available because we can start synthesizing large data sets across large environmental gradients. Okay, so what I'd like to do now is kind of shift towards the second question. I'm going to pull back the global ternary plot that you're seeing here on the left-hand side of your screen. And I want to examine this interaction between DOC and DON in a bit more detail. I think a lot of people, including myself, this is not necessarily surprising that as DOC increases, DON increases. They're two components of the same DOM pool, so we expect them to be correlated. But what we can do now is ask the question of how variable is the relationship between DOC and DON. And what I want to make sure that, just clarify here, what we're talking about is the CN ratio of the dissolved organic matter pool, this large pool of organic matter. And we know that CN ratios are strong predictors of ecosystem processes. And what we're going to be able to do now, what I'd like to do next is test two fundamental null hypotheses. The first being that proportional scaling of DOC and DIN is invariant. And that second, DOC to DON molar ratios are ubiquitous across watersheds. To do this, to examine DOC-DON scaling properties, we're going to use power law slopes. Power law slopes have been key to understanding fundamental relationships and processes in nature. Some of you may be familiar with these papers I cite there. And identifying these scaling properties can offer clues to underlying mechanisms, which can then help us produce hypotheses which can then be subsequently tested. And so on the right-hand side of your screen, you see a hypothetical figure of logged concentrations. And if we discover an isometric line, which has a slope of 1 when we're in this power law space, there's proportional scaling. What it means is that changes in DON are insensitive to changes in DOC. And this becomes our null model. And just for frame of reference here, I just want to point people to James Brown's paper, The Fractal Nature of Nature, Power Laws, Ecological Complexity, and Biodiversity, which has been an important paper for me to understand the utility of power law slopes. So what we can now explore, though, since we have a null model, a slope of 1, proportional scaling, is, well, what happens when we start deviating from a slope of 1 in this power law space? Slopes greater than 1 indicate relative enrichment of DON relative to unit change in DOC. Slopes less than 1 indicate relative depletion of DON relative to increases in DOC. And then the extreme case would be chemostasis and a slope near 0, where there are no changes at all in DOC concentrations, regardless of how big magnitude changes are in DOC. We can also use this type of approach to explore isometry, or the equality of the slopes among sites. And I've drawn another hypothetical example here of stream A and stream B with equal slopes. And if we detect this situation across watersheds, what this could indicate is that the external sourcing of dissolved organic matter is stable and that CN ratios are not changing, or the internal cycling, the internal controls within the stream are relatively homeostatic, and they're using those portions, those fractions of the DOM pool equally. Now, the flipped example of this is that, well, what if these slopes are different? What if they are unequal? So I've redrawn the slope here, the power law slope for stream B. And you can see in site B, we have relative depletion of DON per unit change in DOC at site B relative to site A. So when we see these types of differences, we can now make inferences regarding the hydrological and biogeochemical controls on the C-rich and N-rich fraction of dissolved organic matter. And just want to note here that this is where collaboration and diversity within the working group becomes really important, because site knowledge becomes really key here to help explain these differences. Okay, so what we're going to do now is we're going to first look at the global scale pattern of how DOC and DON relate to one another. So what we're looking at here is the correlation between DON on the y-axis and DOC on the x-axis. I want to note that these are raw data plotted in log-log space, but I'm showing this figure because we can still look at this figure and look at real concentrations, and we can kind of wrap our mind around what we're seeing. Notice that on both axes, we're spanning 2 orders of magnitude across all our different sites. Each point within a figure is a site mean. And so we see a tight correlation here. And the statistics I'm about to share actually come from the power law regression. And in particular, we used reduced main axis regression. And what this does is we have to log the data first. So I'm showing the raw data here, but now I'm going to share the result of the RMA here. And what we'll see is that we have a slope of 0.89. So what this means is that DON increases at 90% for each unit change in DOC. So this is pretty close to a slope of 1, but we're experiencing what we're observing here is a slight bit of depletion in DON per unit change in DOC. We also have a high explanatory R square here. About 71% of variation in DON is explained by DOC. So now what we can ask, though, well, is how does the scaling properties, so the slope, and the strength of the relationship, the R square, vary across sites? And so we're going to use the same data set here for the most part and explore how the variation in those 2 metrics. And what we did next is we limited our analysis to sites with at least 10 points. They had to have a significant DOC-DON relationship. And we're using a slightly more conservative alpha here due to the large size of many of these data sets. And I'll pull back in this figure so we have it, so we can reference what differences in slopes mean. And then here's what we're seeing with respect to variability in terms of the power law slope and the amount of variation explained. Power law slopes are on the y-axis here. R square, adjusted R square in particular, is on the x-axis. Each point, again, is a site. So what we're looking at, for example, in this site right above the red pointer, is that site's DOC-DON power law relationship and the amount of variability in that relationship. So again, the slope here reflects the scaling property, and the R square represents the variability in the stoichiometry, the stoichiometric variability. The G here, up here in the right-hand corner, is the statistics from the global relationships, just so we can see that in context of the variability at the site scale. So the things I want to notice here is, first of all across sites, everything has a slope of less than 1. However, we have a great deal of variation in slopes in terms of that scaling property. So there are some sites that experience a high degree of DON depletion with increases in DOC. And there are some sites that have a high amount of variability in terms of the stoichiometry of DOC and DON. But what's really intriguing in this figure for me, and something I've been kind of staring at this figure for a long time, is what appears to be a shift in linearity. If you notice in this portion here that I've circled, we have a linear relationship between the scaling property and the stoichiometry. But then somewhere right around about 30% here, all of a sudden we're into a nonlinear relationship. We have this high degree of variability now. This is particularly intriguing for me because what it means here, for example, is that we have some sites that have, sites, for example, right across here that have slopes almost near 1, very identical. But look at the variability, the stoichiometric variability among sites. This is really intriguing, and I can't quite come up with an explanation for this. But this is a lot more variability than I think I originally anticipated. And again, if we go back to the literature, in particular, James Brown's paper from 2002, they indicate that deviations from linearity suggest an increasing number of mechanisms at work. So in the context here of dissolved organic matter, what this suggests is that there are some sites that have really one dominant controlling mechanisms in terms of the stoichiometry of DOM. It's not necessarily the same single controlling mechanism among sites, but that there's potentially a univariate control in these sites here. And then when we step into this nonlinear portion of the relationship, multiple mechanisms are potentially at work and interacting, and multiplicative effects maybe controlling the variability in DOM stoichiometry. But now what this does is this allows us to make these types of hypotheses and go out and test them. And I think we can test some of them with large observational data sets, but some are going to require field work where we can actually manipulate systems to go after some of these mechanisms. But this is a really interesting idea in these shifts from linear to nonlinearity. Now what I want to do is, again, I want to scale down from the global scale that we're seeing here down to kind of the region or the LTER scale and look at isometry among sites. So in this table, what we're looking at is the variability in power loss slopes, one among sites within an LTR region or a biome. So for example, here within the Andrews LTR, this is the mean power loss slope across the roughly 10 different watersheds within the Andrews. This is the coefficient of variation. Next here, one column over, we're seeing the mean power loss slope across seasons in the Andrews and the coefficient of variation. So now what we're able to do is look at how DM stoichiometry varies over space and over time, time being represented by seasons here. And so in the Andrews, for example, this is quite surprising to see more variability across sites than we do across seasons. If I point you here to the third row in the Lamprey River here in New Hampshire, we see the same type of phenomenon, that we see more variability across sites than we do across seasons with respect to DOM stoichiometry. This was particularly surprising because we have such strong seasonality in New Hampshire, variability in organic matter inputs between leaf fall and snow melt. But I expected the variation here in seasons to be quite high. But I think what we may be seeing here is that gradient of development that we saw from the ternary plot expressing itself, driving this different variability in stoichiometry. And if we compare that to what we see at Hubbard Brook, for example, we see in Hubbard Brook the system is flipped. We see more variability across seasons than we do across sites in Hubbard Brook. Hubbard Brook is up in the White Mountains. There's less development impact due to development. So we may be seeing that stronger seasonality here where there's a lot of snow melt too in spring, and the changing organic matter inputs throughout the season are now expressed here. As we would expect in Lochio, there's not much seasonality here, but we do see more spatial variability in the stoichiometry. We can hypothesize that could be due to changes in the forest composition with elevation, potentially some type of interaction with the lithology gradient. But what this allows us to do now is look at spatial and temporal controls in the questions of stoichiometry. And I want to show you just a couple exemplar data sets here. The first, this is from the Bonanza LTR and the Taiga. What we're seeing here, DOC and DON on the y-axis again, DOC on the x-axis. And these values here, 0.52 and 0.38, are again the power law slope. And you can see that the power law slope is steeper in spring, the red points, compared to the power law slope in summer. And I think what we may be seeing here is the influence of hydrology and the spring freshet in particular. And the steeper slope represents more hydrological flow paths being activated and new pools of dissolved organic matter being exported to the stream, including a higher amount of DON. However, in summers, that hydrology and the spring freshet kind of churns off a little bit. We're seeing relatively more DON depletion now, indicating a new dominant source of dissolved organic matter is now being inputted into the stream. We see a similar type of situation from the Finnish data set in those northern boreal forests. See big differences here in the power law slopes. In the northern sites, so again, the red sites here, we have a very steep power law slope compared to a much shallower power law slope in the southern watersheds. So these southern watersheds are experiencing more DOM depletion per unit change in DOC. You'll also notice that a high degree of variability in the stoichiometry within these southern sites. We hypothesize that this is an anthropogenic signal, as most of the development in Finland is in the south. And then lastly, these are the data from the Lamprey River Observatory here in southeastern New Hampshire. And this is the really tight and surprising seasonality signal that we observed. And you'll see here that the power law slopes across, especially fall, spring, and summer, are highly invariant. And again, this is this really surprising result to see the lack of seasonality with respect to the stoichiometry. And then lastly, although it is, like I said, tough to, or challenging to test mechanisms, we can make hypotheses about mechanisms. I want to try and say a little bit about the role of biology in terms of dictating the stoichiometry of DOM here. So these last couple of slides are going to reflect on the role of biology in controlling DOC and DON stoichiometry. And that's what's reflected on the y-axis. So that's what I'd like us to focus on first, is this DOC to DON y-axis here. And what we can do with such a large data set is, for the first time, kind of name a number, point to a number that reflects kind of the global number that is DOM stoichiometry. And from this data set, it's indicated that 31 is the DOC to DON stoichiometry of the dissolved organic matter pool according to this data set. But what I want you to notice again is that we have over 2 orders of magnitude represented here with respect to the CN ratio on this y-axis. What that means is, above the red line, this is very high DOC to DON dissolved organic matter in these systems, highly carbon enriched, or a lower DOM to N content. Below the red line, we're seeing a higher DOM, more nitrogen in that DOM content there. So we're seeing a big shift. Now let's focus on this x-axis for a moment here. And what I've done is I've plotted DOC to nitrate ratios on the x-axis. And this has been used as a way to describe energy versus nutrient limitation of the system. So if we slide out to the right-hand side of this axis, in a high DOC to nitrate area, these are systems where nutrients are limited, in this case nitrogen, and nutrient demand is going to be high. If we slide to the left-hand side of the axis, this is where a lower DOC to nitrate ratio and where energy demand might be high. And I think we can use this gradient here of nutrient demand versus energy demand to understand the ecological duality of dissolved organic matter. And what I mean by ecological duality here is where it serves as a nitrogen source and where it serves as an energy source. Because in these hot systems that have high nutrient demand, this is where we find very high DOC to DON ratios, suggesting that the nitrogen of DOM is being used as the nitrogen source in those systems. As we start to move down this gradient here, this becomes heavily nitrogen enriched, depleted relatively in carbon, and it's in these systems where DOM is now shifting to being a predominant energy source. So again, a way to look and examine the ecological duality of dissolved organic matter and the potential role of biology in controlling that. So let me try and summarize what we've covered here today. One is, I think we've highlighted maybe some new ways of thinking about the pool of total dissolved nitrogen. The ternary plots were kind of this novel way of thinking about the whole pool interacting together. And through this, we have a new way of kind of thinking about ammonium as a currency of transfer dictating the composition of the TDN pool. Through this synthesis and other ongoing work, we also know that dissolved organic nitrogen is potentially more ecologically relevant than previously thought. There are a lot of mechanisms controlling DON, and it may have its own unique set of biogeochemical controls, including hydrological controls, physical controls, and biological controls. And that's where I want to remind us of the figure that was produced by Susanna Bernal that incorporated DON into the nitrogen cycle of stream ecosystems, and that there's this potential feedbacks within the cycle itself. Fourth, we identified a global scaling property of DOC and DOM, and that there's a high degree of variability in the stoichiometry, 2 orders of magnitude across the scaling, with respect to scaling and stoichiometry across watersheds, and a high degree of variability across seasons, depending on which system we're studying. And then lastly, I think this forces us to ask the question, then what are the ecosystem consequences of these shifts in dissolved organic matter in the nitrogen pool and the way they're interacting? How does this affect processes of nitrogen uptake and denitrification, retention times, and exports to receiving waters? And then a couple closing thoughts on future possibilities, since I think we're just starting to scratch the surface of what we can do with these large synthesized data sets. Well, one we want to do, because these are long-term, we can start looking at changes in solute concentration over time. And just as kind of a little teaser, it just got these data within the last 48 hours. But we can really take a large look at how DOC and DON concentrations are changing across the Earth's surface. There's been some nice work over the last few years that shows potentially that there are some changes happening, but this way we could take a large examination of that and just highlighting a couple different data sets that show some interesting patterns. In Finland, in the Andrews LTR, down in Loquillo in Puerto Rico, and some dramatic changes in DON from Bonanza. These are very preliminary, but quite intriguing. We can also look at seasonal trends. Again, heavily focused on DOC and DON at this point, but we can do this for other solutes as well. This figure is showing dramatic seasonality in DOC from one of our watersheds here in southern New Hampshire. I think what this is reflecting in this watershed known as Dowse Cape is connectivity and disconnection from an upstream wetland. We can also look at the effects of land use and across environmental gradients. We can start to look at interactions. And we can also look at how other solutes are responding to these changes as well. And so what we have is a growing database, including base cations, other nutrients, physiochemical parameters, discharge values. So this data set is becoming quite expansive, and I think there's a lot of possibilities here. So what I'd like to do here is share my contact information. If the collaborations, additional information, please feel free to reach out to me. I'd love to share ideas and hear about other people's ideas here. And then need to make sure to acknowledge all the wonderful support that is driving this process, particularly from the LTR program and the Network Communications Office there in Santa Barbara, in conjunction with NCEAS, which of course has a lot of NSF support. This is supporting this working group in particular, and hopefully a lot of future work can come from this. So I will close with a final slide in the way that I began, with pictures of some LTR sites, but expressing gratitude for everyone to attend this afternoon in the many different languages that were represented within our working group. So thank you very much for your time, for being here. I look forward to future discussions and open to questions at this time.
Thanks, Adam. I'm sure everyone is doing a virtual round of applause. That was an amazing, amazing analysis. And great work. And so, folks, if you have questions for Adam, go down and look for the Q&A button in the lower panel, and you can type your questions in there, and Adam will handle them as they come. You're going to see a poll pop up on your screen, and the way to get rid of it is answer the questions. Take less than a minute, and it'll go away once you have answered the questions on the poll. Thanks again, Adam. And it usually takes folks a minute or two to kind of find the Q&A, so we can hang in, and you should see the poll now, everybody on your screen. Okay, and Adam, now there is a question, so I'm going to turn it back over to you to repeat the question and then answer verbally.
Okay, so I think the best system here to do is to read back the question, and that's all I need to do, right? Okay.
That would be good.
Okay. So the first question here is, curious during your talk about the effects of land use and land use change over time. So I'm excited to see that's a direction you might pursue. Can you speak a little more about how you plan to address this? Yeah, so a very kind of straightforward answer to that is we're slowly, we have lat-long information for all these different sites, and we'll be able to pull in land use or degrees of development, all those different ways that we can describe land use and changes over time. And then hopefully start to build some kind of multivariate models to look at how change over time is interacting with changes in land use, and then also changes in environmental gradients as well. For example, I think the Finnish data set is a really nice example of that, where we have gradients of development, which are also against gradients of forest harvesting or peatland percents. So I think that's our plan for that moving into the future. It takes a lot, but we're slowly working our way through trying to pull in all of that information to be able to start doing those models. The second question here is, what was the hardest part in assembling or using the data in your analysis? I'm really glad that question was asked. I will admit that it was a lot harder than I thought. I anticipated some challenges, but the challenges were more than I thought, even though I was trying to plan for them. Really, it's a QA/QC question is the first thing that comes to my mind. There's so many different ways that we measure solutes and we describe solutes, the ways we report units on solutes, tracking changes and how solutes were measured with respect to instrumentation. So for example, some solutes may have been measured with one method and then later changed to a second method, which changes the way we calculate them. So for example, nitrate. Is it nitrate being measured, or is it nitrate N being measured? And be able to track those changes and the variability of in the history of those records is quite challenging. And to have to track all that information down is difficult, because to be able to get those values so we can compare them obviously requires a little bit of math, a little bit of a conversion but to be able to have that history at hand and make sure it's right is a big challenge. And then taking all of these different data sets that are formatted differently and synthesizing them into one merged system or database that we can use effectively is quite a challenge. And again, that points to the massive amount of help the NC's data team has done. So I think that ultimately the challenge is the diversity among data sets, the way things are recorded and reported poses challenges, and something I've been thinking a lot about with respect to how as a community can we have a universal way of discussing stream chemistry and the reporting of solutes. I think it'd be very valuable to have that conversation. Next, could these results change in intermittent rivers? Did you consider this in your analysis? No, we haven't considered that yet. But we could do that in certain systems if we have, as we go back and get the history of discharge records. Clearly, this is going to be an important question in certain biomes, certain Mediterranean climates, and also with potential of increasing droughts. We need to consider how intermittent or ephemeral streams, how their biogeochemical cycling is different. You know, for example, here in New Hampshire, which we think is a pretty consistent, an area that has a pretty consistent precipitation regime. During the summer of 2016, a lot of our streams ran dry for the first time during a pretty big drought. And so I think this is a really important question moving into the future. So with historic discharge records, we might be able to get an initial picture of that and something that would be of a great value. So thank you for that question, Juan. Yeah, they're modeling. So the next question, are there modeling techniques you find promising for projecting future change in solute concentration using long-term monitoring data to inform models? There are. I believe. I will confess that I'm not a modeler, but one thing I am interested in is the ability to do earth casting and project these into the future, which is something I'd really like to see. And we can do that with long-term data sets, I think, pretty well. If we have, in addition to the chemistry, if we have the physiochemical parameters to match those data, we could build some robust models in terms of changes in temperature, dissolved oxygen, or discharge. We could make some nice predictions, I think, through earth casting to look at how solutes will change over time. This kind of goes back to the previous question about intermittent streams. So that's something I'd like to talk a lot more about in the future and starting to brainstorm about, and what I think these data sets can be immensely valuable for. So thank you for that question, James. Next question, have you looked at proximity of your sites to dams? The short answer is no. But we could do that with potentially certain data that potentially we know are above or below dams, or if we have synoptic data, or if people can help identify some data sources that are long-term that have a above and below type of treatment, we could ask that type of question. I'm not aware that we have any of that currently in hand, but I suspect that could be accessible. But that could be a very interesting question moving into the future. Thank you for that question.
Okay, that wraps up the questions. Adam presented his email address at the end of the talk, and I'd like to remind everybody that these webinars are recorded and they're placed on the LTR Network Communications Office website. So please share them broadly, and I'd like to, it's a little bit awkward, I know, on a webinar, Adam, but I'm sure people are just clapping in their offices for such a educational and fantastic talk.
Thank you.
So we will close that down and hope that folks can join us next month, same time, same place. The schedule's on our website, and thanks again to the attendees as well.
Yes, thank you everybody for being here.
Okay.